My name is Terrence Markin, and I'm the Executive Director of the Graphene Council, the largest community in the world for graphene producers, researchers, application developers, and end users. We have hosted the Graphene in Healthcare and Medical Applications conference series, and the video you are about to watch is from that series. So our next speaker is Jeffrey Draa, who is the CEO of Grolltex. Grolltex is a longtime member of the Graphene Council and one of the really good innovators in this space.
Thanks very much, and thanks everybody for thank you, Terrence, for setting this up. Terrific opportunity. We appreciate it very much. My name is Jeff Draa. I'm CEO and co-founder of Grolltex. Grolltex is short for Graphene Rolling Technologies, and we're going to talk about that quite a bit. My topic today is scaling production toward adoption of 2D materials for biosensing. A couple of words on the company. So we are in San Diego. You may categorize us as a UC San Diego spinout, close to the campus, still have many programs with the campus and the school. All of our operations are here in our clean rooms in San Diego. A final word, we consider ourselves the 2D materials scalability company. We don't work on anything that isn't scalable. We also believe we're the largest commercial provider of monolayer graphene and HBN in the States today. And then final word, clarity. So today, we're only going to talk about CVD graphene. That's large area monolayer graphene as grown by CVD. That's the only material that we produce. So if we're going to talk about what's needed to scale this material for biosensors, we actually have to kind of cover three areas. Number one, let's see if we can talk about making the material, the base sensing material, graphene, inexpensive, large scale, high quality, sustainable, etc. Number two, we have to be able to make devices on top of that graphene or with that graphene kind of in the same mode. It has to be inexpensive, high throughput, high yield, repeatable, etc. And then number three, and this area is often forgotten, but just as important, the packaging. Let's make sure we can make the packaging inexpensive, flexible, biodegradable, multifunctional. So we have to be able to do all three of these things to really talk about making graphene biosensors scalable. So let's talk about the first area first. So we stand on the shoulders of giants, as many of us do. What you're looking at here is the depiction of the discovery in 2010 by the two teams, one Korea, one at University of Texas. This is, of course, the breakthrough of making graphene on a large-scale basis via CVD. So you're looking at high-quality, large-area sheets made via CVD. The problem and the reason that this process isn't more proliferated, we believe, is in the next step down, which is transfer. So once you've made your high-quality sheets, large area on copper, in this case, the next step is to transfer that graphene onto the surface of interest. And in this depiction, what they're showing here is acid etching the copper away. Acid etching copper, of course, takes many hours. It's a very, very dirty process, creates a lot of very nasty effluent. Also, you destroy a lot of very, very expensive high purity copper. So this is the issue that has kept proliferation of CVD large-scale graphene. Critical breakthrough here in the manufacture of graphene with this one remaining problem, the transfer issue. And this is why we started the company. My partner and co-founder, Dr. Zaretsky, while he was a researcher at UCSD, came up with a process to remove graphene from copper and reuse the copper. So we do not destroy copper here. As it happens, the more times you use the same piece of copper, the better and better quality graphene you get. So that's a nice side benefit. Our process can be done in minutes versus the hours it takes to acid etch the copper for transfer. And the equipment needed to do our process is off the shelf, has existed in industry for decades, very well understood. So we believe we have an important piece of the puzzle of unleashing massive graphene production at low cost. So very quickly, this is one of the more technical slides. This is the depiction of our patented process. It starts with the assumption that you've grown graphene on copper, and this could be any size. This is an arbitrary size. The next step is to metallize that graphene. So in this case, we're putting on nanometric nickel or cobalt. And the reason we do that is that the graphene has about a 7 to 10x adhesion strength to this new nanometric nickel layer as it does to the copper underneath. So it wants to stick to this new nanometric nickel more than the copper. Then we peel that graphene off of the copper. The copper goes right back into the beginning of the line to regrow more graphene over and over again. In this depiction, the next step is taking the graphene, rolling it onto a PET sheet. Then the next step down is getting rid of this nanometric nickel. And in the end process, you've got your sheet graphene on PET for a finished product. For example, flexible displays, OLEDs, etc. Might want to use it for that. A simple look at this might say, well, hold on a minute, Grolltex, you're just changing one metal issue for another by putting on this nanometric nickel. Anyone who's worked with these materials will tell you that getting rid of 20 nanometers of nickel is quite trivial as compared to trying to get rid of microns of copper. So this is our patented process for transfer of graphene. We've been employing it here successfully at Grolltex for 5 years. You need 3 pieces of equipment to do our process. Basically, one piece of equipment, of course, CVD growth. So this is for creating or synthesizing the graphene. Next step is metallization. Many different tools available to do metallization, sputtering, also can be done via CVD. And then the third process is patterning and transfer. Patterning if you desire that, but thin film transfer, those are the pieces of equipment you're looking at here. These are very well understood processes. And this equipment's been around for a long, long time, all of these here. And in fact, for all the equipment we're looking at, we have been to the suppliers, the vendors who design and make this equipment to make sure that our processes and our recipes can work on all of them. So we're pretty satisfied that when it comes time to ramp up for large area need, we're ready to do that. That's why we started the company. That's what we do here in San Diego every day. As it happens, our factory operates only at about 10% of our capacity because that's the market need. So we stand ready to ramp up our facility for any kind of use cases that come along. We're going to talk about a specific use case here in just a minute. So to kind of wrap up that piece number 1, we believe we have the pieces here to make the base sensor material industrializable, large scale, sustainable, and scalable. Okay, let's move on to the second area, number 2. Let's make the sensor also inexpensively and high performance configured. How can we do that? So let me go back to the transfer process here for just a minute. And let me point something out, something interesting. What if we stop right here, and instead of etching away this new metal layer, what if we do something with that metal layer? That metal layer can be patterned and different devices and device structures can be made out of that metallization layer. Here what we've done is pattern that nickel, put some copper on top, cap it again with some nickel, and that can be many other metals as well. And this pattern happens to be set up for a large scale OLED. So this could also be used, of course, for solar panels, flexible displays, etc. Very simply, these are straight square patterns, but this could be made with some slight jagged shapes, and in that case could be made of flexible OLED material as well. So all kinds of interesting things can be done with by leaving this new metallization layer on and patterning it. And for purposes of our discussion today, we can talk about what can be done with regards to roll-to-roll patterning, which is what you're looking at here. Several vendors have roll-to-roll lithography and patterning tools. The feature size can be down to tens of microns, which is pretty much where you're going to see many biosensing applications today, those kinds of geometries. We have patented processes in place here for making multimodal sensors with the schema you're looking at. We have also a patent series for cardiotoxicity sensors. It's a whole nother area we can talk about another time. And then more recently, we've been asked to pattern and create sensors capable of detecting viral RNA and proteins, et cetera, all done with the schema that you're looking at here. So our conclusion is that graphene structures are roll-to-roll patternable with this kind of a schema. Again, very simple, every piece of equipment been in industry for decades, and we have done this in-house. I'm going to talk about results of that here in a little bit, some of the results. So that kind of covers number 2. Let's move on to packaging. Can the packaging for what you've seen here, can those devices be packaged in a flexible, inexpensive, biodegradable manner? So today, not only do we make materials, but we also help some of our customers with further steps down the line. And here's kind of what I mean by that. We have over a dozen customers who use our material on silicon wafers to make biosensors, okay? And they're biosensors in all kinds of different areas. We have customers that use our material to make sensors for genomics, CRISPR chip, for example. We have customers that use our material to make sensors that detect glucose for diabetics. We have customers that use our material to make protein binding interaction sensors. So all kinds of different modalities, but one of the common threads is that they all operate on silicon wafers. Well, we did a deep dive into our customers' uses and designs, and we found a couple of things. Number one, none of our customers make particularly small devices. In fact, the geometries are all measured in tens of microns, so not particularly hard devices to make. Number two, none of our customers use the silicon on the silicon wafer as part of the plumbing of their device. All these devices reside on top of the silicon wafer, and the wafer is basically just a processing tray, okay? Another thing is that if you're going to make your device on a silicon wafer, you're fairly restricted in the kind of package that you can put that in. So some of our customers have like a thumb drive type device, for example. Others use more like a finished kind of a packaged chip. But the long and the short of that is between the cost of making your device on silicon and then mounting it in this fairly expensive package, you end up with a fairly expensive biosensor. That's a real cost from a real customer. And with many applications, that's just fine. But we took a dive into using some of our processes here, some that I already kind of went over, and we thought, well, maybe we can make these devices on plastic instead, and sure enough, we can. And not only does making devices on plastic dramatically reduce the cost, but it introduces a whole another area of applications that are possible if you make your graphene-based biosensors on plastic, and it can be biodegradable plastic as well. Now, let me show you one slide of kind of what I'm talking about. So these were the first 3 form factors that we came up with for graphene-based biosensors on plastic. And this first one here is a bubble pack. So this is the same blister pack. Go to the drugstore, get your aspirin or whatever, pop the pill out. We talked about a graphene-based biosensor inside that blister pack, and the blister performs kind of 2 functions. Number one, it obviously protects the sensor environment till you're ready to use it. But number 2, you can pre-charge that blister environment with some functionalizing chemistry, getting ready for the test of your choice, right? You can program this thing months in advance. Now, the reason we came up with this was a customer of ours came to us and said that he had a customer who was a farmer, and the farmer wanted to go up and down the rows of the crops of his field, and he wanted to test for the health of his crops. And at the time, our customer could only offer a $50 thumb drive kind of device. And of course, that plugs into a laptop. So here's the farmer. Every time he uses the thumb drive, it's a one-shot, throw it away. That's $50. And he has to carry the laptop with him in the field to plug the thumb drive in. Well, instead, if he can pull out these blister packs made of biodegradable plastic and pop one off, and it costs 30 cents, and he uses a little handheld reader, then that much more closely approaches a reasonable use case for him. So that's form factor number 1 that we came up with, is this blister pack. Form factor number 2, so imagine in this case, what you're looking at is a 10 by 10 grid. So this is 100 of these graphene-based sensors in plastic. It's very similar to the blister pack, slightly more rigid, but this lends itself to, in this case, the 96 well plate automation for a high-scale study that many large pharma and other companies want to do in the tools that they work with. And it's not just limited to 96 well plate. This could be made any size. It could be 1,000 sensors on a sheet if you wanted to. Finally, number 3, what you're looking at here is a multiplexed microfluidically plumbed graphene array chip. This is a little plastic chip. This one happens to have 5 sensors on it, but we can pretty easily double to triple the density of that. And this could be a viral diagnostics chip. So you could program each of these channels, one for different virus, right? One's Zika, one's MERS, one's SARS, one is a control channel. And here you've got a very inexpensive, and again, we can make these things for pennies each here at our facility. In fact, the crude one you're looking at here, we did make all here plastic and everything. So this can be made as a diagnostics chip. So we have some unique cost and form factor advantages working with plastic, and it opens up all kinds of different markets and applications in biosensing as opposed to operating on silicon. Now, when we first went down this road of graphene biosensors on plastic, this had never been done before. If you Google graphene biosensors, and many of you, of course, know that if you do that, you're going to see thousands of published papers of all kinds of sensors with this material in all kinds of different areas. Virtually all of that work, graphene biosensors, virtually all of it is on silicon. And it makes perfect sense why it would be. If you're going to make some nano device with some new material, go to the silicon infrastructure. It's there, nano printing and etch deposition, it's all there, right? So that makes perfect sense. So the work had not been done. Does graphene-based biosensors work on plastic at all? So this was our first go at answering that question. As it happened, we picked, well, okay, let's pick some important issue to solve. Let's design a graphene biosensor on plastic to find glucose. In this case, we happened to use glucose in the concentration of saliva. It took us actually less than 2 weeks to design and test this initial glucose sensor on plastic. It worked very well. That was about 1.5 years, almost 2 years ago now. Since then, we have proofed it out also with in a JDP with a large pharma company. We proved out that graphene biosensors on plastic work with protein binding interactions. We showed it with them in 7 different disease areas that they were interested in. We've also in-house proved out that graphene biosensors on plastic work for virus detection. In this case, it was molecular detection. We detected SARS DNA, it's actually RNA in the concentration of saliva, and our sensor picked that out in the first 73% of the signal, I think, was picked out in the first 16 seconds. So the conclusion here is that graphene biosensors on plastic do work. That's our experience. We have that internal data. And we're very excited to roll this out with our customers. We are now working with our customers that operate on silicon. Many of them are very excited to make the transition to on plastic, and we're starting that process with them now. So kind of the conclusion of my talk here is that our experience is that these 3 areas can be covered. We have been working on this now for a couple of years. That number 1, we can make the base sensor material inexpensive, high yield, high throughput, sustainable. Number 2, we make the devices in the same way. And then number 3, to wrap it all up, indeed the packaging can also be made cheap, flexible, multifunctional, and in a biodegradable manner. Thanks very much for your time, everyone. Happy to take questions now. You can reach me at the contact information that you have there, phone number and email. And Terrence, I'll sit back. I guess I'll let you sort of take the questions and formulate them and send them in if you like.
Excellent, excellent. Jeff, thank you so much. Really appreciate that. I tell you what, it's very impressive when you think graphene was only really isolated in 2004. The Nobel Prize awarded in 2010, so 11 years ago for that, and was still very much a research oddity. To come this far this fast is absolutely amazing, and I hope it's not lost on everybody. This transfer technology to be able to get graphene onto a plastic substrate is huge. The other thing that should not be lost on anyone is to recognize that this isn't theoretical. This isn't something that maybe graphene could work for this, but you've demonstrated and are demonstrating in multiple use cases, real world today, being used for these medical analytics devices, which I think is really important to see. And I am quite confident that there will be a lot more to follow for you. I do want to kind of do kind of a context check. You know, when Laureline, we asked her to open up today's conference to kind of get the perspective of the customer, the end user, from the medical devices and diagnostic equipment and other applications perspective. How closely, because you're obviously working with companies, customers, how closely does what Laureline outlined as the priorities and objectives map with what you're hearing from your customers?
Yeah, you're right. It is early days with regards to this material and biosensing. There are a few areas that are further ahead than others. Two of the areas that are that I would say have at least 3 to 4 years of good, solid data are the genomics field. So using graphene in gene editing, and then number two, protein binding. So a couple of our customers have been working in that area for proofing out protein binding for observing reaction of new drugs, of course. So those two areas kind of have the most. Now, more recently, of course, because of the pandemic, there's been a real press on virus detection. So several new customers to us popped up and wanted to start using our material for making handheld virus detection and different modalities, right? So there's a couple of guys in antigen, there's a couple of guys in molecular. The molecular area, I think, has the most promise with regards to virus detection. And in fact, we were asked by a couple of our partners here in San Diego. So San Diego, of course, on the La Jolla shores, there are a few large research institutes. We had some programs going with a couple of those partners. One, for example, in exosomes to detect cancers. And those large partners at the beginning of the pandemic, those large partners came to us and they basically just cleared their desks. They said, look, what we were working on with you guys is very interesting. We need to put it on hold. We want to work with you on a COVID sensor. So this was back when early days when everybody was, again, all hands on deck, right? And so we started working with a couple of these research institutes. And the very first road we happened to go down was this detecting virus via molecular means. And that's when we came up with that on plastic sensor chip. And the results were, they were very quick. There was no question about what we were getting. And as it happened, we were working with molecular or viral RNA in this case, and that also went well with our sort of scalability mission, if you will. We don't work on anything that's not scalable, and making a graphene biosensor for DNA RNA was really easily scalable, at least with what we had in-house here, our patented capabilities. So that's kind of the area that we marched down and got some very good results with that so far. Looking for a large diagnostic company partners, by the way, right now, just to let you know that we would rather partner with a large diagnostics company to try to get these things out more quickly. And this platform that we come up with for detecting virus with graphene biosensors on plastic is quite programmable for the next virus and the next and the next. So there's nothing special about COVID and this sensor. It's quite capable of other viruses and bacteria as well. So I hope that answered your question.
No, it certainly answered the question. And I want to go back to an earlier question and combine it with some of the others that are coming up. Just what is the role that the graphene plays with these analytics, and how to discern between one target molecule or one target chemical and another, and combine that with another question. Have you had any results for, and I'm not going to refer to this correctly, but ctDNA or microRNA in blood, right? Have you used it for any analytics for out of blood serum? And yeah, I guess that would be the question. So is it the graphene that's actually doing the detecting, or is it you're adding molecules and using graphene as a critical component?
So that's a really good question. Let me take the second question first with regards to blood. I'm aware of one of our customers who is very, very early days using our material to try to detect on blood in that area. So that's not an area we have a lot of background with yet. But I believe there is a fair bit of published information in that area. But again, we haven't seen yet. Now back to the first question. So with regards to the graphene, there's actually many, many ways to use graphene to get a biological signal. And let me kind of boil it down to its most basic kind of premise. So with graphene being the most conductive substance known at room temperature, and with it being very, very, very thin, this material, when it's in the sensing environment, in any sensing environment, if there are electrons running across graphene, if there's any kind of disturbance, and that means an atom, a molecule, an RNA strand, any kind of biological activity on top of the graphene, while there's electrons flowing across it, that change in resistivity is a very clear signal. It picked up very quickly, very robust manner. So those are kind of two of the properties that make this material ideal for biosensing, that it's very, very thin and that it's very, very conductive. Additionally, graphene is very biocompatible. Biology loves to sit on graphene. Since it's also transparent, then a sensing schema, and we did this internally as well, can be over-engineered to not only collect the electronic signal I described, but also an optical signal, since it's transparent, right? So you can do all kinds of neat photonics things at the same time. Now, the last thing you mentioned about functionalizing graphene, it is possible. In fact, we have some patents here internally and have done a fair bit of work with decorating graphene in one instance with gold. Now, this will enable some really terrific sensing capabilities in the area of, for example, a DNA and RNA, because then these probes love to anchor on the gold. So gold on top of graphene enhances many sensing modalities by way of configuring the graphene for different outcomes, right? So all kinds of things to do with graphene, many ways to enhance the sensitivity and sort of fine tune what you want to pick out.
Excellent. And I do have an additional question for you. This might be our last one. We have about 3 minutes before the top of the hour. One of the questions from the attendees is about the mobility. And you just mentioned graphene being highly, highly electrically conductive, the most electrically conductive material at room temperature. So the question was, what is the mobility of graphene that's transferred via your MAE process? High mobility graphene is important in this particular application, would be for optical electronics. And question was, is it better quality, quote unquote quality, than conventional roll-to-roll product? How would you describe it? And I know that's kind of a sensitive area, but
it is, and I'm gonna answer it unfortunately a little bit generically, but that's okay. At least I hope that's okay. So there are a few quality questions that come up now and again with regards to our processes, not just synthesis, but also transfer, when the question becomes, well, is this good enough quality graphene for a given application? So far, we have not had a customer in any area, whether it's biosensing. By the way, we have other areas of activity. We have photonics sensor customers. We have customers who make all kinds, advanced audio equipment, et cetera. We have yet to have a customer come to us and say, the quality of the material you're giving me doesn't work for my use case. Please make higher quality material. Now, we can do that. There are several knobs we can tune here to make higher quality graphene. In other words, a larger crystal, et cetera, et cetera. Now, that would make it more expensive material. And so far, all of our customers have said, no, I don't want more expensive material. What you're providing is fine. So to broadly answer the question, so far with regards to our synthesis and our transfer, we're meeting all customer expectations with regards to quality, and no one has said, I want more expensive material. So that has not been the case yet.
Got it. And Jeff, I think we're going to leave it there. I know there's a couple more questions, but you have your contact information in your slide deck, which we will distribute. And I encourage anybody who wants to learn more to reach out to Jeff. Obviously, he is deeply knowledgeable in this area, and I want to say congrats again to Grolltex for the progress that you have made, and we're very proud to have them as a member of the Graphene Council. On behalf of the Graphene Council, I want to thank everybody for participating and for your interest in this sector. Clearly, graphene has a very important future in healthcare applications, whether it be for devices, medical sensing, diagnostics, or even in some of the infrastructure of gowns and devices and elastomer products, etc., that can be enhanced with graphene. So with that, I want to thank you very much. Thank you for your interest in the Graphene Council. If you have any questions, if you need any guidance, if you need information, anything to do with graphene or any of the companies that you saw in the past 2 days, please reach out to us, and thank you for your time and attention.